Okay, so we're going to review some terms and talk about a study that was presented in this lesson. It's a study about red pandas, who I think are so incredibly cute. I just love them. I used to volunteer at the Sacramento Zoo, and while I was there, they had baby red pandas in the back. They weren't even on display, and they were just so cute little fluff balls. So I'm including lots of pictures. Okay, so let's review experimental design because it does get brought up in this lesson. I think it's an important thing to remember, and it's reintroducing the variables, the terms for the variables. So remember, in an experiment, there's something that made you question, and now you're going to test, and you're going to come up with a hypothesis for that question, and then you're going to make a test to test that hypothesis to see if your possible explanation is correct. And the classic example I always give is like the leaves changing color in the fall. The question is, what causes that? And then you can come up with a hypothesis. Again, your guess, what you think is the answer. And let's say we think our answer is the change in temperature. Then you can create an experiment to test your hypothesis. Is it the change in temperature? When you set up an experiment, you're going to have different variables in that experiment. You're going to have an independent variable. Every experiment has to have an independent variable. That is the thing that you're going to change. That is the thing that you think is causing something. So you're going to change it and see if it is having an effect, if that change is having an effect. So you have the independent variable, the thing that is changed. And in our tree example, it could be the temperature. So you can test trees at different temperatures and see if that causes their leaves to change color. Which brings me to the dependent variable. The dependent variable is the thing that you think the independent variable is affecting, and that's the thing that's going to be measured and recorded. So with that example of the trees, if we're changing the temperature, putting trees in different temperature chambers, and we think it's affecting the change in the color of the leaves, then we're going to measure and record how many leaves change color at each temperature. So that's the dependent variable. The independent variable is the thing that was changed in the experiment that is different between the different setups. And the dependent variable is the thing that we're measuring and recording. We're thinking that the thing that we're changing is causing an effect on the dependent variable. All other variables need to be controlled for. Everything else that we can control for are the control variables. So anything in my example that could affect the dependent variable, the leaves changing color, needs to be controlled for. So maybe, the amount of sunlight, the type of soil, the amount of water, the amount of oxygen, any other like predators or herbivores in the area, anything else that you can control for needs to be controlled. Now, when you create a graph, there are rules on where you put the independent and dependent variable. The independent variable is supposed to go on your x-axis, and your dependent variable goes on your y. Okay, your lesson, and I thought this was kind of cool, I've never heard this one before, gives the acronym DRY mix to help you remember where they go. So DRY has a D and a Y in it, so dependent variable goes on the y-axis, and mix has an I and an X in it, so the independent variable goes on the x-axis. And here's a couple graphs that we can look at. So, we've got light intensity on the x-axis, so they're changing the light intensity, and then they're measuring rate of photosynthesis. So in this experiment, light intensity was the independent variable, rate of photosynthesis is the dependent variable. In this experiment, so we're looking at typing speed, we've got weeks of practice, and then words per minute. So they are measuring and recording the words per minute, and they're taking that measurement at different weeks. And I mean, the assumption is that in between, these people are practicing, right? Maybe 30 minutes a day or something. And what they are seeing is that through time, speed goes up. Okay, so you do need to, you're going to get a question on the quick check about which one is the, I think it's the independent variable. So think about which one is being changed, which one can be changed by the experimenter, and which thing can be measured and recorded and looked at. Okay. Okay, so let's review genetic terms because that gets brought up in this lesson. I think reviews are always good. So we've learned a lot about genes. Remember, a gene is a segment of DNA that codes for a protein, and proteins are things that do stuff in your body. All right, they are what determine your traits. You can have different alleles for the genes. So you have a gene that determines your eye color, but there are different alleles. You can have brown eyes, blue eyes, green eyes, and variations in between. So the alleles are those different variations, and in our examples, we use different letters like capital letter, lowercase, same gene, different allele. Genotype are the combination of alleles you have for a gene, and you could either be homozygous, homo meaning same, so you have the same two alleles, big A, little a, or you could be heterozygous, hetero meaning different, so you're going to have two different alleles, big A and a little a, or a big B and a little b. Now you have two copies of each gene, two alleles, because we have homologous chromosomes. Right? Homologous chromosomes are the chromosomes that have the same genes. They could have different alleles, but they have all the same genes on them. And because we are diploids, we have two copies of each gene, so we have two of each chromosome. And those are referred to as homologous chromosomes. So you have two of chromosome 1, and these are the homologous pair or the homologous chromosomes. For chromosome 2, which has a bunch of different genes on it, and chromosome 1, you get two of them, and those are the homologous chromosomes. You get one copy from mom and one copy from dad. Total, you have 23 pairs of chromosomes, 46 total chromosomes. Right? When you create gametes, you only put one set. So you put 23 total in one of your gametes. Right? So the homologous, so again, homo means same, meaning they have the same genes. Those are the homologous chromosomes. The term that gets brought up that we haven't seen before is locus. And locus is the location of a gene on a chromosome. So you can think of it as the address. So you would say this gene is located on chromosome 2P. So one branch, one side of the chromosome is referred to as P, the other side is Q, and then there's going to be a number. So the locus is just the address where that gene is located on the chromosome. What chromosome? Is it located at the top, middle bottom, very bottom? Okay. Another new term that gets brought up is single nucleotide polymorphism, or SNP as we call them. And these are mutations that only change a single base in a DNA sequence. We've heard that before, right? You're like, oh, hopefully you're like, oh, that sounds a little familiar. Yeah, it's a point mutation. We were calling it a point mutation last time. This lesson, they're calling it a SNP, and you can call them both. Okay, so SNPs are point mutations, and they're used a lot to look at, actually we can use these to help identify people by genetic markings, because the ones that happen in non-coding regions build up through time, and that's where you can find differences in people. But in the areas that actually code for individuals, there's small changes. Okay, your lesson has a claim. It says that when the environment changes, you see an increase in SNPs in a population. So it's saying that when there's an environmental challenge or change, that there's an increase in genetic diversity because the organisms are adapting, is basically what they're saying. And that when there's no change in an environment, individuals remain very similar to one another, so you don't get a bunch of genetic diversity. And this is completely wrong. And I am blown away that somebody wrote this in a lesson. I put a note into our curriculum communication and I have not heard anything back. Like, am I misinterpreting? Are you saying something a different way? Like, anyhow, that's not true. If anything, an environmental change is going to decrease genetic diversity because we're causing selection. We're now selecting for individuals who have a specific trait. And the ones that don't have that trait, they're being selected against. So their genes that cause that trait are going to decrease in the population. And if anything, because mutations happen normally, those SNPs, those changes, they happen all the time. Environmental changes are not going to increase the rate of those changes. Okay, environmental changes, unless you're like shooting someone with like x-rays or dousing them with like chemicals to cause mutations, environmental changes like deforestation, habitat change, ecological, any of these ecological disturbances that we've talked about are not going to cause mutation. If anything, they're going to select for individuals who already have mutations to withstand that change. So there's a question on your quick check that says, which of the following scenarios would reduce the number of inheritable traits in a population, causing individuals in that population to remain very similar to one another? The answer, what you should check, so you get the point, is a desert that has existed for millennia, because this is the only one that doesn't have any change. Everything else is like, there's a big change in the ecosystem. But that big change is not going to cause mutations to occur. So anyhow, hopefully it gets corrected for next year. It blows my mind. The lesson also talks about the panda studies. Now we get back to seeing cute pandas. I want to explain this a little bit. So they looked at different populations of this panda, so A, B, C, D, and they're telling you the population size. They never explain what this number of segregating sites is, and I'm not sure what they're referring to, but these are really big numbers. And then it reports nucleotide diversity. And if you look at these numbers, you see that A, C, and D, their numbers are very similar, where B, which is a smaller number, almost half the size, is very different. The lesson goes on to say that, they determine that use this information to determine that there's different species of red pandas, which is true. You can do that by looking at genetic diversity. Genetic diversity can show you that there are different species, and there's different rules to make them different species besides genetic diversity, but they're calling this population B the Himalayan pandas, and the other ones, the other three, the Chinese pandas. It does say that the Chinese pandas have had more environmental issues, and that's why there's more genetic diversity. Again, not true, where the Himalayan pandas have been very stable, their populations have been very stable, and so there isn't much genetic diversity. Again, not true. These numbers are obviously made up. It also shows this graph later on in the questions in the practice section, where they looked at the Himalayan pandas through time. So now they're looking at different generations. So this is our independent variable, the time, and then they're measuring genetic diversity and seeing that it's changing. Oh, it's going up. Okay. And because I think they're just so cute, your code word for this video is red panda. Your code word for this video is red panda. Okay, so those two graphs I just showed you, the first one and this one, are in the lesson. Well, this graph shows up in the quick check, and there's a couple questions about it. And this graph is kind of like bringing it all together, like I said, incorrectly. But in this graph, we're looking at the Himalayan pandas and their population over time. So we're looking at generations, our independent variable. One of our independent variables is time. They're looking at the average mass of the red panda. So their size is not changing. The average number of panda cubs born to a mother each year. So that would be the average. So it's 32432 changes, but no big trend in change. Looking at nucleotide diversity, so that data that was on that other graph, it's going up. looking at acres of deforested land. All right, and that's going up. Some big changes. These two, it stayed the same, and then it went up to 450, now 4,000, now 54,000. Big changes there. And then population size, which, oddly, is not changing. So this is another issue I bring up. If you're hacking out land, if you're destroying habitat, it's most likely that the population size is going to decrease. So I find that a little odd. Maybe it wouldn't, but most likely, especially if you're, taking out 54,000 acres of land, you would expect that there's going to be a decrease in the organisms that live in that habitat. But whatever, it is whatever. There's a question about this. So there's two questions. One asks you to identify the independent and dependent variables. Remember, the independent variable are the things that you are changing. So the generation, so the time that you're making the recordings is an independent variable. If you're making recordings at different times, that's your independent variable, okay? Another thing that you could argue is a change is the amount of deforestation that's occurring. That's something that they're changing. Everything else is something that's being measured and recorded. So everything else would be a dependent variable. And they can look to see if generation time is affecting any of these things or deforestation, if that's affecting it. There's another question about this graph that says, which explanation for the increase in genetic diversity of the red panda population is supported by this data? And the answer that they're looking for is that red pandas in the population have encountered new environmental challenges presented by the increase in deforested land, and this is selected for different adaptations over time. Again, if you have selection, your genetic diversity goes down because you're getting rid of the other stuff. Anyhow, that's the question that the one that they want you to put, even though it's incorrect. But now you can get the point for it and hopefully know that that is incorrect. Okay, the other thing that gets brought up in this lesson is sickle cell anemia, and this is a really interesting one because it is the mutation that causes sickle cell anemia is a positive mutation in some way. So first off, anemia is a disease, any type of sort of disease that affects your red blood cells getting oxygen to your body. So your red blood cells, that's what they do. They bring oxygen to your body, and if that's not happening effectively, then you have anemia, you feel weak and tired, and there could be other symptoms associated with that, but those are some of the main classic ones. And there's different things that can cause anemia. In this case, my image isn't showing up. Weird. In this case, it's sickle cells, which the cells are in a different shape. So red blood cells, oops, why is it not letting me draw? So red blood cells typically are disc shape. So circular, they're a little indented. Sickle cells, they're in that sickle shape. It's caused by a point mutation. It's an autosomal recessive point mutation. There's a change in the hemoglobin, which is the protein that carries the oxygen. And the point mutation, the change in one amino acid, causes the hemoglobin to have a different shape. And so the overall blood cell has a different shape. Now, people with this, so it's autosomal recessive, so you can see 3 different types of genotypes. So homozygous dominant, big S, you don't have sickle cell, okay? You don't have sickle cell anemia, you don't have any sickle cells in your body. If you're heterozygous, so you have one big S, one little s, you don't have sickle cell anemia, but you are a carrier of the gene, of the mutation, meaning you can pass it on to your offspring. If an individual is homozygous recessive, so little s, they have the disease. You have sickle cell anemia. So your red blood cells aren't carrying the oxygen as well, so you have the anemia. And then another issue with it is the sickle cells can get clogged in your blood vessels, and that causes tissue damage and other issues. Historically, people with sickle cell anemia do not live as long, and sometimes not even to reproductive age. That's getting better with modern medicine, but historically, if you have sickle cell anemia, you're kind of weak and sickly and you don't live as long. Now, the thing that's interesting, individuals who are heterozygous, they have some disc-shaped cells and some sickle-shaped cells. So, but they have enough of the regular disc-shaped cells that they don't have the anemia. They don't feel sick, but there is a difference in their body. And that difference makes them resistant to malaria, right? They don't fully understand why yet. They think it has to do with maybe the malaria parasite can't attach to the sickle cell protein as well, or possibly, and also that people who are heterozygous and have the two different shaped cells, they filter their blood more quickly, so the plasmodium that causes malaria can't stay in there as long. But for whatever the reason, we know that people who are heterozygous, that has at least one allele for the sickle cell shape, is they are resistant to malaria. They can get malaria, but they don't usually get it as bad. Now, in areas where malaria is prevalent, you see a lot of individuals being born with sickle cell anemia because the people who are heterozygous have an advantage. So if you are heterozygous, you don't have sickle cell, so that's not making you sick and weak and die, and you can't get malaria compare as bad. You're less likely to get sick and die from malaria compared to the homozygous dominant individual. So you have a better chance of surviving. You have a better chance of surviving. You have a better chance of reproducing. And if you reproduce with someone else who is also heterozygous, because they've been able to survive better, then your offspring, you have a 25% chance of your offspring having sickle cell anemia. So you see more babies born with sickle cell in areas where malaria is prevalent because you have that selection pressure, because people who are heterozygous have an advantage. Now, if you live in a region that does not have malaria, like the US seems like a little, on this graph, a little standout because we kind of have a higher amount, but we don't have babies being born with sickle cell. That's what this graph is showing. We do have a higher amount, but we don't have malaria in our country. So people who are heterozygous here in the United States do not have that advantage because we don't have that selective pressure. So there's some other answer to that, why it's common. Could it be immigration? I mean, I don't know what the answer is, but it's not because those with malaria have an advantage. Okay, but it's kind of a cool example, and there are other examples out there, but I'm sure this video is getting pretty long, so we'll go ahead and end it. Other examples of advantageous mutations like that. So if you have any questions, please let me know, and thanks for watching.